Thank you for giving me the opportunity to present some of our data. My title sort of infers that I'm going to be talking about more than one granulomatous lung disease. In fact, we do study various T-cell mediated granulomatous lung diseases. I'm only going to focus today on chronic Brillian disease, which is where I've spent most of my career working on, but we are doing analogous studies in pulmonary sarcoidosis. So these are very much related diseases. They have a similar clinical presentation, similar sort of outcomes that can go all the way from just sort of a spontaneous remission to fibrosis and the worst case scenarios of respiratory failure. So chronic Brillian disease is a sort of a classic example of a gene by environment disease. You have workers who have a disoccupational exposure to beryllium. It's a lightweight, highly valuable metal in industry. Number 4 on the periodic table, used in various, as I mentioned, aerospace industry, other high-tech industries. Over a period of decades, years to decades, these workers become sensitized to that metal by inhalation, working, machining the dust, inhaling it, lodging in their lungs, and they become sensitized to beryllium, which is defined as having a positive proliferation test to beryllium salts in vitro. And then among those who are sensitized over a period of years, some of them then go on to develop beryllium disease. That's characterized by granulomatous inflammation that's actually in the lung. It's been well appreciated now for greater than 2 decades that there's a very strong genetic component or genetic association to disease, and that's with the MHC class 2 molecule. These are HLA-DPB1 alleles that have a glutamic acid at the 69th position in the beta chain. And HLA-DP2 is the most common of those GLU69 containing alleles. So chronic Brillian disease is T-cell mediated. It's characterized by a CD4 T-cell alveolitis. And then the hallmark of the disease is the formation of non-caseating lung granulomas. There's an accumulation of beryllium specific CD4 T-cells. These proliferate in response to beryllium in association with specific peptides. These beryllium-specific cells secrete Th1 type cytokines, and they recognize beryllium when it's presented by the GLU69 DP alleles. So this is a cartoon of that interaction. And so we have a CD4 T cell that's expressing an alpha beta T cell receptor, and it's recognizing a beryllium that's being presented by a classical antigen presenting cell that's expressing the alpha beta heterodimer, which in this case is I'm showing us DP2. And beryllium is part of the ligand, but the other part is the part that we don't know, which is what is the peptide. So that's what I'm going to talk to you about today. So this is a flow chart of how we discover antigens in chronic Brillian disease or in any other disease, basically. We obtain bronchoalveolar lavage fluid, and then we flow sort those individual CD4 T cells into 96-well plates and do a single cell PCR separately of the T cell receptor alpha genes and the beta genes. So the goal here is to obtain complete T cell receptor alpha beta pairs. And then from among those, we look at the repertoire that's from the target organ in this disease and decide which ones are our candidates, which ones do we want to study. And to do that, we look at the receptor pairs and look for hallmarks of disease relevance. And so these have been long appreciated and studied over the 20 years that I've been working in this area. You often look for preferential V gene usage, oligoclonal expansions, and then importantly, homologous TCRs, which means that you have different clonotypes, but they express T cell receptors that are similar to one another, in particular in the CDR3 area, which is the part of that T cell receptor that comes into most intimate contact with the peptide within the MHC class II binding. Having looked at the alpha beta pairs and determined which ones we want to study, we construct T cell hybridomas. These are immortalized cell lines that will express our candidate T cell receptors, and then we determine if they're beryllium specific. And so the hybridomas, they respond to antigen by secreting IL-2, and so we have antigen presenting cells. We add beryllium, we see if they respond or not. And you can see in this example that one of the hybridomas is not beryllium specific, but we're pretty good at picking out the ones that will have the antigen specificity we're interested in. The next step, which is I'll go into more detail, is that we use combinatorial positional scanning libraries. These are synthetic libraries that they're peptide mixtures that are organized in a particular way that we can dissect what are the preferred amino acids at particular positions of the peptide, and then from that try to go to the actual epitopes that are stimulating these T cells. So in this study, we had 7 patients. 4 of these were directly ex vivo samples of bronchoalveolar lavage cells. 3 of them were T cell lines, so they were cultured briefly in vitro in the presence of IL-2 and beryllium to further expand what we think are the beryllium specific cells. We sorted these number of cells with the goal of obtaining around 100 alpha beta pairs, which we thought would be enough to give us a sense of what the repertoire is, if there's oligoclonal expansion and so on. So we did that, and this is an example of a public T cell receptor that was found in 6 of our 7 patients. And so each line of this represents a single T cell receptor, in this case beta sequence. We also have the alpha data that I'm not showing you. And so for each patient, you have this clone, for example, 1041, you have this clone that expresses this V beta, this J beta. This is how many times it turned up in our sample and whether we made a hybridoma from it or not. And the key point on this is that within the CDR3 beta, again, that portion of the receptor that comes into contact with antigen, you have this conserved lysine at this position highlighted in blue. And in addition, you have these other amino acids that are sort of repeated. Another way of looking at it is shown here, which is called a TCR web logo that highlights the V beta, J beta usage and the usage of amino acids of each position, and with the height of the amino acid representing the overall frequency of that amino acid at that position. So we're calling this the LKGGG motif. And we made a number of hybridomas, and sure enough, the data on the right here shows IL-2 production in response to beryllium, showing that these are in fact beryllium specific. And we rely, in this case, the antigen presenting cells were splenic B cells from a HLA-DP2 transgenic mouse. We rely on those APCs to provide this endogenous antigen, this peptide, to complete the ligand. We don't know what it is, but it's clearly there because we see a positive response. So I'm probably going to run out of time fast, so I'm not going to really explain the libraries too much, except that just to say that we end up with because there's 10 positions in the decapeptide and there's 20 amino acids, we end up with 200 mixtures. So for example, in sublibrary 1, we focus on position 1 of the peptide and we fix it, either with all 20 amino acids, but in separate samples or mixtures. And then when you actually assay the hybridoma, and this is our standard assay, you can see if that hybridoma, if that T cell receptor prefers that amino acid at that particular position. And if it does, it will respond and make IL-2. So this is an example of a single hybridoma responding to the library. So we have IL-2 on the y-axis. We're looking at 4 positions of the peptide. And what you see is in position 5, if that mixture has a aspartic acid there, and all the other positions are randomized, you get a nice response. So this T cell receptor appears to only want an aspartic acid at position 5. Similarly, at position 7 and 8, glutamic acid is highly preferred. You oftentimes don't really see this kind of spiky result. It's often much more muddy, and you have to work to figure it out. So kind of leaping ahead again, we chose to make a biased positional scanning library, where we then fixed an aspartic acid at position 5 and a glutamic acid at position 8, and then scanned the remaining positions. And in this case, I'm showing you an example of position 2 with the 20 amino acids. So if you think about this, the number of, and then these X's are randomized, these mixtures are much less diverse because there are 2 positions are now fixed, or 3 positions, and you should see a lot more activity. So in this case, and we'll just focus on this upper right because this is the P2 library. So in the context of D5E8, these 2 hybridomas are showing very similar response patterns because they have related T cell receptors. And you can see there's preferences for either cysteine, phenylalanine, and so on. So you get this information for all of the different positions in this biased library, and then you end up doing a biometrical analysis. And that basically takes all that empirical data and creates a scoring matrix. And then that matrix is applied to all possible overlapping decapeptides in a UniProt database, or any database. In this case, we looked at a UniProt human protein database. And then that ranks those peptides using that matrix based on their ability or their predicted ability to stimulate the hybridomas. And so they're all ranked, and we choose some number of them to screen. And in this case, we looked at over 100 different peptides. And when we do that, and we actually could test those peptides on all 8 hybridomas we made, expressing that LKGGG motif, these are the sort of the top contenders at this point. And you can see that this BM64 peptide actually stimulated IL-2 production in all the hybridomas. And then this BM67 in 7 out of the 8. So what are these peptides? These are peptides derived from the CC motif chemokines 4 and 3. So looking at those in a little more detail, these inflammatory chemokines, these peptides are highly related to one another. They only differ at 3 positions, and these are all relatively conserved changes. Of course, they have this D5E8 because that's part of the motif or the bias library that we use. And what we're seeing here is that these hybridomas are cross-reacting to both of these. These are not separate specificities. So we've identified that in those 6 patients that we studied, and we wanted to ask the question at how broad is that T cell response? And so to do this, we used an MHC class 2 tetramer. And so that's shown here as a cartoon on the right. It's basically that DP2 molecule made as a soluble version, and then the peptide, the chemokine peptides, are covalently attached to the N-terminus of the beta chain. And then the whole complex, well, first of all, it's multimerized to account for the low avidity or affinity and avidity between TCR and its ligand, its class 2 ligand. And then it's soaked in beryllium to make it a complete molecule. So in the limited amount of time, I'm going to show you one of the best ever kind of tetramer staining with class 2 MHC staining in a human subject. This is patient 8845. These are bowel cells taken directly ex vivo and stained. And you can see that in this case, the CCL4 tetramer, over 20% of the CD4 T cells coming from the bronchoalveolar lavage of this patient recognize that tetramer. Similarly, the CCL3 tetramer, 25%. Now, these are not independent populations. Remember, these are T cells that are cross-reacting to both. although I can tell you from studies that I'm not going to show, we do have T cells that only recognize the CCL4 epitope or only recognize the CCL3 epitope. Another neat thing that we can do with these samples is combine that tetramer staining with intracellular cytokine staining. And in this case, we took those ex vivo bowel samples and stimulated them with beryllium for 6 hours prior to tetramer staining, and then do the intracellular, in this case, with gamma. And so you can see that there's the two quadrants up here, which are totaling over 30% of the bowel CD4 cells, are making gamma in response to beryllium. There's none made in the control. And that a good fraction of those are actually making or binding to the CCL4 tetramer. And this is the same data of the CCL3 tetramer. So just a quick summary of some of the patients we looked at. Generally, we're looking at about a 3, 4, 5%, although there's huge variation. This is on a log scale of the number of beryllium patients, they're HLA-DP2, that are binding to these tetramers. In this case, the control were the beryllium sensitized patients who haven't developed disease yet. So I'm going to a few minutes. So the one thing I want to say in concluding about this human work is that this data provides a link between beryllium-induced activation of innate immunity. So I'm not really showing you, but beryllium acts as an adjuvant in these patients, inducing chemokine secretion. And so beryllium's not only inducing the protein, but then that very same chemokine is being used as part of the adaptive immune response. So there's this link between innate and adaptive immunity. And the other thing I want to say is, so beryllium disease, I didn't emphasize enough earlier, happens over a period of decades. And people have their disease sort of waxes and wanes in terms of the CD4 T cell responses that you find in the lung. And so we think it's possible that infections might result in flares, disease flares, because when you're infected by some kind of lung pathogen, a virus or something, part of that response is going to be to secrete these inflammatory chemokines, CCL3 and CCL4. And in effect, what that does is it increases the amount of antigen that's now available for those memory CD4 T cells to become reactivated. It's just a thought. So just in a couple, I'm just going to spend 2 or 3 more minutes talking about some of our studies. We have a mouse model of beryllium exposure in an HLA-DP2 transgenic, and the model is a sensitization boost, and then the mice are harvested where they're exposed to beryllium oxide intratracheally, and then they're euthanized at 3 weeks. The mice develop mononuclear cell infiltrates and an adaptive CD4 T cell response. And for something that happens in 3 weeks, modeling a human disease that spans years, decades, the CD4 T cells we do pull out of these mice recognize the same beryllium-dependent ligands that we find in the human subjects. So we added, I meant to add, we added this LPS exposure because this is going to induce chemokine secretion. So it's only one-time exposure. And you can see if you expose them at day 14 and you look at bowel fluid at day 15, you see a big increase in the amount of CCL4 and CCL3 that are secreted. And then you also, by ELISpot, so this is another way of looking at antigen-specific cells, secreting interferon gamma and IL-2, you see a big increase relative to those mice that just received beryllium alone in the number of antigen-specific cells. And what are those antigen-specific cells? Well, they're cells that bind to the CCL4 tetramer. So you see a big increase in terms of the number of cells responding to this epitope, this beryllium-dependent epitope. So we have developed a more severe model of disease in this case. But if we knock that, and this is sort of quantitating that difference in tetramer staining. But if we knock that gene out, So conversely, we take the DP2 transgenic, breed it onto a CCL3 knockout, then we reduce that number. And this is interesting because, and you can see here, the percentage of tetramer binding cells here goes way down. And even though that population of T cells doesn't encompass the entire population of beryllium-specific T cells that are present in the lung, in the mouse or human, it does greatly diminish disease. So here you see in a normal DP2 transgenic, the aggregates of cells in the lung, and here they go away. So the last slide I'll show is that, okay, maybe we can target those cells. And in this case, what we're considering making, we have made, sort of is a CAR T cell that expresses this DP2 molecule with the peptide and beryllium, and use this. So the CAR it's a second generation CAR with CD28, CD3 zeta intracellular domains, and we can use this to target those beryllium specific T cells via their T cell receptor, and look at possibly ablating them and seeing what the effects are in the mouse model. So this project, and a lot of details left out, and this project and the other projects in sarcoidosis were done with lots of collaborators, kind of a team science approach that spanned for many years at this point. And so I want to make a special shout out to Andrew. I was in his lab for a long time. And now the lab is the Falta ATIF lab, or as he prefers to call it, the ATIF Falta lab. And we couldn't have done this without any of these collaborators. So thank you very much.
So Mickey, with the CCL3 knockout, what about CCL4? If they're cross-reactive, is that still generated? So
the CCL3 knockout is hypomorphic for CCL4. I should have mentioned that. So, because the argument would be, well, you have a CCL3 knockout, but the CCL4 is still there, and the antigen is still there, shouldn't have any real effect. Well, CCL4 is greatly reduced in this mouse also. So what was your question?
So it's the question about why wasn't CCL4 picking up the slack from the.
And that's why, because it's not there. And yeah, and I use the CCL4 tetramer here in the CCL3 knockout. So it's much reduced in these mice. Not totally gone though.
You mentioned a couple of times that it's similar to sarcoidosis in some way. So where's the similarity and why?
Well, the granuloma is very similar. And then so I'm a PhD, I don't ever see these patients, but I know that sarcoidosis is a diagnosis of exclusion. And so one of the things they have to do when they're diagnosing it is making sure they haven't been exposed to beryllium. So I think the pathology of the granuloma is very similar. Now, one of the studies we're doing is in Lofgren syndrome. Those aren't really seen, as far as I can tell, in the United States that much. We get samples from our collaborators at the Karolinska, and those have a more distinct phenotype. And those are characterized by CD4 T cell expansions of V alpha 2.3 T cells. And they have sort of a different clinical presentation. So they're a little bit different, and they're one of the groups we've been studying and made progress on.
Is it still a lot beryllium exposure industrial?
No. So the good news is beryllium disease with occupational controls is far less of a problem now, which is good for everybody except those who study the disease. But the occupational controls are reduced. You do continue to have a lifelong risk of disease because you never completely remove, it's lodged in your lungs, it's sequestered in these granulomas, it's still there. But we're basically moving into more of studying sarcoidosis, which is a more prevalent disease. But problematic because we don't know what causes it.
Last question. So this approach of antigens discovery, in this case, or peptide discovery for presentation, other HLA-related diseases where the HLA connection is firm, and has this been used in that? For example, I'm thinking of
All right.
gluten sensitivity, rheumatoid arthritis.
We have hybridomas in the freezer that same approach. I did it as a postdoc. We didn't have nearly the tools. We didn't have access to these libraries. The way we identify alpha beta TCRs, even what I showed you here, is completely outdated. We can now get thousands of alpha beta TCRs through like the 10x genomics platform. But nonetheless, we have hybridomas that are from DR4 patients where we could do this very same approach.
Yeah.
And I'd love to do it if I had the time, for the funding.
Thank you very much.
Thank you.